Okay, welcome back. This is the first of 2 lessons on enzymes and medicines. You might have heard of a molecule that's found, a hormone that's found in human bodies called insulin. It's made by pancreatic cells and is important in blood glucose. Insulin is a hormone made by human pancreatic cells that's important for blood glucose control. Some people, especially those with type 1 diabetes, can't make insulin. So many people with type 1 diabetes need to have regular insulin injections because the insulin will be denatured if it's consumed orally. In this lesson, we'll look at the various structures shown on the left, which this is an example of an insulin protein molecule, which relate to the type of bonding within the protein molecule. So here we're looking at enzymes and medicines, part 1. So we're going to look at the different types of protein structures, the principles of enzymes, and the specific effect of temperature on enzyme function, including denaturation. So to start with, amino acids are the monomers or the building blocks of protein. There are 20 different 2-amino or alpha amino acids, which are the building blocks of proteins. And they all have a carboxyl group, an amino group, and because carboxyl is always the highest priority, this is carbon 1, so this is carbon 2, and the amino group is attached to carbon 2. This here is the side chain, which is sometimes given the symbol Z or sometimes given the symbol R. And the difference in the side chain is the difference between the various amino acids. So a 2-amino acid is an amino acid whose carboxyl and amino group are bonded to the same central carbon atom, which is carbon number 2. Now, of the 20 amino acids, 9 of them are what we call essential. So they need to be supplied in our diets because our bodies can't synthesize them. That's just a bit of background before we start talking about proteins. So the first level of protein structure. So the primary structure, there's 4 levels of structure, and the primary structure contains covalent peptide bonds. Now, we know covalent bonds are very strong, and if we have a look at the structure of human insulin, it contains 2 chains. We can see a pink chain and a blue chain, and the pink and the blue stand for the various amino acids. They're given symbols for their biological name. Then we've also got some sulfur bridges that link across. We'll talk about the sulfur bridges later. At the moment, we're just looking at the sequence of pink and blue amino acids. Just like the necklace that I was wearing a second ago, the various beads in this necklace represent the different types of amino acids or the building blocks. So I've got quite a large amino acid here. I've got another one here, a smaller one here. All these are varying by their R chain or Z chain, and we've got various beads that join up. That sequence of beads, the order that they're bonded in, is the primary structure. We can see that the arrangement of the amino acids is like the order of beads in a necklace. So if we look at this example, we have the amino acid residues. That's what's remaining of the amino acid after they're bonded together in a condensation reaction. What we end up with, we lose that hydrogen, we end up with peptide bonds between the amino acid residues. Now, peptide bonds are also called amide bonds. Amide bonds is a bigger category. Not all amide bonds are peptides, though. Only amide bonds in biological molecules, in biochemistry. So because we're talking here about protein in the human body, when we're talking about insulin, that is biochemistry. So we can call these bonds between the amino acid residues, we can call them both amide bonds and peptide bonds. So they're formed from the condensation of amino and carboxyl groups on adjacent amino acids. And the atoms involved are CONH. And we can write that as CONH. And then we've got the rest of the molecules on either side. So the primary structure is the sequence of amino acids in the polypeptide chain, So this is our polypeptide chain, just like beads in a necklace. The next level of structure is secondary structure, and it's how the necklace is rearranged in 3 dimensions so that the beads from various parts of the necklace might interact with each other. And that contains hydrogen bonds that can be easily denatured. That's in contrast with the primary structure, which contains really strong covalent bonds. So here we have an alpha helix, which is like a spiral. And what we've actually got are hydrogen bonds between adjacent amide groups. So here we have an amide group or peptide group, another amide group, and in between we have a hydrogen bond. Now we know that hydrogen bonds are intermolecular forces and they're much weaker than covalent bonds. So we've got another hydrogen bond here and another one here, and they come about where adjacent amide groups on a folded necklace where they come close to each other. So the hydrogen as part of one amide group bonds to the oxygen as part of another amide group. So adjacent amide links. And we're now starting to get the necklace to start folding up on itself. It's weaker than covalent bonding, and because it's weaker than covalent bonding, it's more susceptible to temperature changes than the primary structure. It's very hard to disrupt the primary structure. Another type of secondary protein structure is an arrangement which is called a beta-pleated sheet, but they both contain hydrogen bonds. The next level of protein structure is the tertiary structure, and all types of bonding are involved here. So we can have disulfide bridges, which are covalent, and they come about from SH groups on adjacent amino acids that contain sulfur. Not all amino acids contain sulfur. We have hydrogen bond in tertiary structure. We have ionic interactions, which are very strong. And we also have weaker dispersion force interactions between non-polar side chains on amino acids. So we have lots of different types of structures in the tertiary structure. So we've got the sequence of beads, which is the primary structure. We've got that folded onto itself, held together by hydrogen bonds, which is the secondary structure. And then this whole thing then has more shape, more three-dimensional space carried out about it because of the tertiary structure. So the strong covalent disulfide bridges are between 2 adjacent cysteine residues. So this is part of cysteine and this is part of cysteine. We've got our strong ionic bonding between NH3 plus and COO minus groups. We've also got hydrogen bonds, as we do in the secondary structure, and dispersion forces. So there's lots of different types of bonding going on. Because we've got weak dispersion forces and slightly stronger but weaker than covalent hydrogen bonds, it's relatively easy to disrupt the tertiary structure. So the tertiary structure is able to be changed by temperature. Not all proteins have a quaternary structure, but those that do, it's where you have more than one tertiary structure that's bonded together. And an example is in insulin. Insulin actually exists in the body as a hexamer. That's 6 insulin units that are held together with a zinc ion in the middle and has similar bonding to the tertiary structure. So disulfide bridges, ionic bonds, hydrogen bonds, dispersion forces. In this case, it's also an ion dipole bond with the zinc ion held at the centre of the 6 strands. But not all proteins have a quaternary structure. Okay, now we want to look at some principles of enzymes. Enzymes are really useful because they're biological catalysts. Now, being biological, they only operate within a very tight temperature and pH range as opposed to inorganic catalysts like nickel metal. So they're highly specific for a particular reaction, but they're also very susceptible to being denatured. Think of an enzyme as a 3-dimensional structure. It might just be one part of it that is the active site. And the active site is the region that binds to the substrate, which is the part that starts off as the reactant that is catalyzed. They only function within a specific range of temperature and pH. Once an enzyme is denatured, it no longer acts as a catalyst. And when we're talking about denatured, we mean that there's disruption to the secondary, tertiary, or quaternary structures. So the primary structure, the sequence of beads in the necklace, stays the same, but the overall three-dimensional shape has changed, so the active site no longer exists. Now, because enzymes are catalysts, we can look at energy profiles, and we can see the progress of a reaction on the x-axis and the energy in kilojoules per mole on the y-axis. With the activation energy without an enzyme is all the way from the heat of reactants to the top of this activation energy complex. But with the enzyme, we get a lower activation energy, and that's how an alternative pathway is provided. The way enzymes work, there are various models, but the model you need to know for VCE is what we call the lock and key model, and it's commonly used to explain how enzymes bind to the substrates. The three-dimensional shape of the substrate exactly fits the active site of the enzyme. So here we have the enzyme, and this is the active site here, and here are the substrates. This can insert, and they exactly fit, just like a key in a lock. And then once the shapes match up, then the catalyzed reaction can take place. So the enzyme substrate complex is where the substrate is bound to the active site of an enzyme. So let's look now at how temperature affects enzyme function. So this graph here shows that enzyme activity varies over a temperature range. And in this case, the range is 0 to 60 degrees Celsius. So this could be a human body enzyme, for example. So on the y-axis, we've got increasing enzyme activity. And we see it increases from 0 degrees Celsius up to an optimal temperature, which is the maximum, and then decreases thereafter. So we have low activity below the optimal temperature, and a little bit above the optimal temperature, we start to denature, permanently disrupt the active site by altering the secondary, tertiary, and quaternary structures of the enzyme or the protein. For most warm-blooded animals, the optimal temperature is 37 degrees Celsius, which is our body temperature. However, different animals have different optimal temperatures. So animals that live in the Arctic, for example, and thermophilic organisms that live in high-temperature vents under oceans, they have lower and higher optimal temperatures for their enzymes because they operate in a different environment. But for humans, the optimal temperature is 37 degrees Celsius. Let's have a look at what happens at lower temperature. At temperatures lower than the optimal temperature, we know that temperature is a factor in rate of reaction, according to collision theory. So in this region here, we've got lower activity because the temperature is lower. And that means that or we can say as temperature increases, kinetic energy increases. Therefore, there's a greater chance of successful collisions with energy equal to or greater than the activation energy. And that in turn leads to greater enzyme activity up to the optimal temperature. And did you know, as a rule of thumb, for an increase of about 10 degrees Celsius, there's a doubling of reaction rate up to the optimal temperature. At temperatures greater than the optimal temperature, enzymes are permanently denatured. So it doesn't matter what you do, you can't get that specific secondary, tertiary, and quaternary structures back once they've been denatured. It's because you've disrupted the dispersion forces, the hydrogen bonding. You might have also disrupted some of those disulfide links, ionic bonds, and the structure is permanently changed. You can think about when you poach an egg, the raw egg white is colourless and translucent. When you heat, the egg white turns into a solid mass. You can't uncook that egg. The primary structure in denaturing remains unchanged. That's really important. But the active site is no longer functional because you've changed the secondary, tertiary and quaternary structures at temperatures greater than the optimal temperature. Let's have a look at a worked example. Explain the different types of bonding that are found in the primary, secondary and tertiary structures of enzymes. So if this is worth 3 marks, we need to say something about each level of structure. The primary structure of enzymes involves strong covalent amide bonds. We could also use the word peptide if we're talking about enzymes, which are biological molecules. So that's 1 mark. The secondary structure involves weaker hydrogen bonds between adjacent amide groups. So they're the CONH groups. So that's the key feature of bonding there. And the tertiary structure involves many types of bonding, including strong disulfide covalent links, strong ionic bonds between adjacent cation and anion groups, weaker hydrogen bonds between adjacent polar groups, and weak dispersion forces or even weaker dispersion forces between adjacent nonpolar groups. So that's where our 3 marks come from. Okay, let's summarise what we've learned. The primary structure contains strong covalent amide bonds, like the beads in a necklace. The secondary structure contains weaker hydrogen bonds between adjacent amide links, which are the CONH groups. The tertiary and quaternary, if there is one, the structures, include a range of bonding types, including strong covalent disulfide links, strong ionic interactions between cation and anion groups, weak hydrogen bonds between polar groups, and even weaker dispersion forces between adjacent nonpolar groups. And remember that not all proteins and enzymes have a quaternary structure. We can use the lock and key model to explain how enzymes catalyse biological reactions. The enzyme has a specific active site which matches to the shape of the substrate to produce the enzyme substrate complex and catalyse the reaction. We can look at the enzyme activity over a range of temperatures. We have our optimal temperature. We get the maximum enzyme activity produced. At a temperature less than that, we have lower activity due to collision theory. We have a lower rate of reaction if the temperature is lower. The temperatures much greater than the optimal temperature, we denature the protein and it no longer functions as an enzyme or for its particular shape. Next, we've got Enzymes and Medicines Part 2. So I'll see you then.